Hi, I'm Samantha Moreau, biology class, and for my homework number 1 assignment, I chose to do a cell. And I wanted to prove that science can not only be interesting, but delicious. So here is my cell. And I'll explain to you a little bit about the components that I used to create the cell and what these things represent. And you'll notice that the entire cell is coated with this thin plastic layer, and that represents the plasma membrane. It encases the cell, so yes, it is there on purpose. So before I put my cell down, we can go ahead and observe the nucleus. I have used an egg here because it adequately depicts the layers and the detail that I wanted to show. The yolk being the nucleolus, the white being the general nucleus, and I even left the shell intact to show the nuclear membrane, but you really can't see it. I didn't anticipate that some of the detail in this would be as hard to pick up, so I will actually go ahead and show you what I've used to make up the rest of the cell. But you're going to have to take this part for what it's worth, because the nucleus, the heart of it all, well, I ate the other half, but this is where chromosomes and DNA are stored, so my wickedly bad sense of humor, there's the blame. Right there in the nucleus, the heart of it all. Now, before I put my cell down, I will show you the cytoplasm. It's this yellow material. It basically fills the cell. A lot of activity goes on there, but everything that's yellow, I used pineapple jello, and it is the cytoplasm. We'll talk a little bit about smooth and rough endoplasmic reticulum. I represented the smooth endoplasmic reticulum using a gummy worm, and the rough endoplasmic reticulum I used with a gummy worm that has been rolled in a green sugar. Now, what's the difference between these guys? One needs a shave, one doesn't, right? Wrong. The difference is that the smooth endoplasmic reticulum, along with not having ribosomes attached to it, is actually there to synthesize fats. And the rough endoplasmic reticulum is designed to sort proteins and create complex compounds. So similar, yet different in appearance and different in function. Now the ribosomes, the little guys that were clinging to our endoplasmic reticulum and making it rough as opposed to smooth, well, those guys actually manufacture proteins. And they do one of two things. They either roam about the cell as little transient ribosomes, or they cling to the endoplasmic reticulum. I've represented those on my cell as these little granules of sugar, but I guess the scientific part of me failed to recognize that they would melt and dissolve partially as the gelatin set in the refrigerator, so it's the green component that you can kind of see, but a little bit got lost in the translation. Okay, next we have our centrioles. There are typically about 2 per cell, and I represented those in my cell using these little black licorice. And these guys are responsible for separating chromosomes during cell division. Next we have our mitochondria, and our mitochondria are organelles that convert energy from nutrients into ATP, and I represented those using 3 little jelly beans. Next we have the Golgi apparatus or body. I represented that with a bit of blueberry fruit roll-up, and it is actually layers of membrane, and they make compounds with proteins and they prepare them for transport either into or out of or within or out of the cell. Next we will talk about the lysosome, and the lysosomes are pretty incredible little buggers. Here we find out that our digestive system is not where all the digestion takes place. Our lysosomes, which I have represented with little red sixlets, they actually contain digestive enzymes and material in the cell digested right here. It all happens, this little mini digestive system within each one of our cells. Amazing? Amazing. Next we have our peroxisome, which I've represented with a little golden raisin. And it actually houses enzymes and is used to break down harmful substances in our body, like drugs and alcohol. Can't do without these. Now I can also add that for our lysosomes and our peroxisomes, when we talk about terms of homeostatic balance, if one of these guys isn't doing what they're supposed to do, really bad things can happen within our bodies. It is known to be blamed for autoimmune deficiencies and disease. So they may be tiny, but they are mighty. And last but certainly not least, we have our vesicles, which I have represented with orange sixlets. And those I push to go as far to the outside of my cell as possible because those guys are actually little pods. Those storage containers you see sitting in people's front yards, that's how they work. They store materials in bulk, move them in and out of the cell all at once. So they're very efficient, and they believe in less is more. So now that we've talked a little bit about our cell and the ingredients I used to put this bad boy together, we can take another look at it, and hopefully anything that gets lost in the translation with film, you got to see up close and personal, so we can figure out that all of our ingredients are there and intact. So here is our cell with our plasma membrane covering, our yellow cytoplasm, our nucleus, our smooth endoplasmic reticulum, this bloody mess representing our rough endoplasmic reticulum, and of course our little melted ribosomes within the cell. Here our centrioles. We also have our lysosomes, and we have our vesicles, and we have our peroxisome way down in there, and then over here we have our poor Golgi body, which isn't faring so well, and deep down inside we can see our mitochondria, our jelly beans that are floating, and if you can't see them, they're there, trust me. So this is the layout of a typical animal cell. So I hope that this has given you a little bit of an understanding of how our cells are put together. It is so amazing. I cannot believe that somebody figured all this out and all we have to do is learn it. It's pretty amazing stuff. And now that I think about it, I think I'm going to take my cell and put it back into the refrigerator, wait for my daughters to get home. You know what they say about science? Research, always creating more questions so that we can have more research. So I'm hoping that they will dig into it and ask themselves the burning question, why did Mom put an egg in my Jell-O? Thanks for watching, and have a great day.
Still looks strange. Mom?
Yes?
Why is there an egg in my Jell-O?
Why indeed?